Now here you see we are dealing with a new chapter that is known as sexual reproduction in flowering plant, and here we'll begin with the angiosperm. Now basically what are angiosperm? Angiosperm belongs to those group of plant that bear flower. That means all flowering plant that includes the monocot and the dicot comes under this angiosperm. So once we need to go into the detailing of the chapter, we need to know the morphology of these flowering plant, or rather we need to know the morphology of a flower. Now here you see you can see this diagram. Now here it's a generalized diagram that is common to all the flowers. Now we know that the structure of a flower has been divided into two whorls. One is the accessory whorl and one is the essential whorls. Okay, so what is accessory whorl? Accessory whorl means they are circlets. The flowers are arranged in circular manner, and once we go in from outside to the inner side, the sepals and the petals include together as the accessory whorl. Why they are known as accessory whorl? Because they do not directly take part in the reproduction. Now once and inside the androecium and the gynoecium, that means the stigma, you see the stigma, style and the ovary, this includes the whole female unit. It may be known as a pistil or it may be known as a gynoecium. Okay, and here you see the anther and the filament. Now together all the anther and the filament, you see they are numerous in number and they includes the androecium or the male gametophyte. Okay, so these are the structures that are essential for sexual reproduction in a flower. Okay, now you see now here you see once we shall be dealing with the sexual reproduction in a flowering plant. Three basic categories has to be known here. One is the pre-fertilization, second is the fertilization, and the post-fertilization. Okay, now the pre-fertilization here is the topic that we will be dealing with today. That means pre-fertilization includes the gamete formation or the pollen formation in case of a flower. Now you can see the structure. This is actually the anther, and that is borne on a filament, and the anther is connected to the filament by the connectives. Okay, in general the anthers are bilobe structure, and you know this structure, you know, it is made up of it is an inner layer is known as the tapetum, and these are the tapetum that bears the tissues for this pollen formation. Okay, now you see the pollen, or we call it as a microspore. Okay, now we have microsporangia, that is also known as the microspore mother cells, that divides by the process of meiosis in the ratio 1:4. That means one cell produces 4, that means tetrad. Those are the actually pollen cells, they are haploid. And if we take of the sporogenous cell, that is the tapetum, we have 4 layers. Okay, what are the 4 layers? You see epidermis, endothecium, the middle layer and the tapetum. Okay, now the structure of the tapetum is very essential and morphologically as well as cytologically important, because those cells of the tapetum gives to the sporogenous cell which undergo meiosis to produce the pollen grain. Now here you see this is the structure of the anther. So this is the pollen structure. Okay, now what is this? You see here the lines of desiccation. That means when these gets dehydrated, you know, the anther lobe actually burst in a longitudinal manner. So that is what known as a line of desiccation. Now here you see we have a thing that is known as diothecus. What is diothecus? Now diothecus is actually two-lobed structure. Okay, now you see we have 4 tetragonal structure. It's a bilobed structure of an anther and is very distinct in its transverse section of the anther, and the anther is 4-sided tetragonal structure. Now I have already said that microsporangia that is located at the corner of the two lobe. The microsporangia develops further and become a pollen sac. It extends longitudinally all through the length of the anther. See, longitudinal means here, longitudinally, okay, and the structure of microsporangium, now it is a transverse section of a typical microsporangium near the circular in outline. See here, if you cut it, it is circular in outline. Okay, see, and the innermost layer, I told you earlier also, the innermost layer is known as a tapetum. And what is tapetum? See here, it nourishes the development of pollen grains. The cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Okay, now see, tapetum, the younger-most layer, it's compactly arranged and it is a homogeneous, and that is known as sporogenous tissues. Okay, so microsporogenesis is the process that we are going to deal it with detail. Okay, so the anther develops, the cell of the sporangium tissue undergo, see, meiotic division. Okay, it's meiosis, okay, to form microspore tetrad. Here you see the diagram. Here you see outer, I already said it is the epidermis, then the endothecium, okay, then the sporogenous tissue, then comes the tapetum, see, middle layer, okay, and these tapetum tissues, you see, they have microspore mother cells, endothecium, epithelium. These microspore mother cells, you see, they undergo meiosis. Okay, now see, each cell of the sporogenous tissue is capable of giving rise to the microspore tetrad. See, one tetrad formation, 4 cells together, joined together, and this process, see, you need to underline this, the process of formation of microspores from a pollen mother cell, PMC, through meiosis is called the microsporogenesis. So if you are asked what is microsporogenesis, so basically microsporogenesis is a process that leads to the formation of a pollen cell. Okay, that means pollen mother cells give rise to 1:4 is the ratio. The microspore as formed, they are arranged in clusters of 4. I said the arrangement is tetrad, so it is known as a microspore tetrad. And if see, again the process of desiccation is also a very important, and it is also hormone related. Okay, the anther, when the anther matures, then it has to get dehydrated. The microspore desiccates from each other and develop into pollen grain. Now, inside each microsporangium, several thousands of microspores or pollen grains are formed, and they are released with the desiccation of the anther, and that is how the pollens are released. Now, you can see the structure of pollens. You see, here it is tetrad, a single pollen structure. There is a nucleus and a vacuole to it. Okay, now how does the pollen germinates when it reaches the stigma? That would be the next part when we'll be dealing with the fertilization.